We all know this field is in constant change, and it's defined and pulled and twisted and redefined. And our next speaker, Dr. Emmanuel Ruiz, will discuss how omics data is changing our molecular applications. Dr. Ruiz was awarded a PhD in molecular and cellular biology. And throughout the years, she was confronted with the need for computational biology tools in order to answer biological questions. And she had to acquire an expertise on bioinformatics and biostatistics. Dr. Ruiz, can't wait to hear what you got for us. You're on stage. Can you turn your video on, by the way? I cannot hear you or see you, Dr. Ruiz.
Here we are. Hello, sorry.
There you are.
Let me try again. All right.
Yeah. I can hear you fine. And if you want to go to slideshow mode, that would be great. There we go. You're
it's
on
perfect.
Your own.
Thank you.
Good luck.
Thank you. My name is Emmanuel Ruiz, and I am a senior postdoc bioinformatician in the Veterinary School of Louisiana State University. It's an honor today to present my work experience from the bench to the computer and how bioinformatics with omics data have drastically changed my career path. So I was born, raised, and got my education in Lyon, France. I completed my PhD in the Cancer Research Center of Lyon, and the main purpose of the research project was to decipher the interaction and the collaboration between the epithelial mesenchymal transition regulators with oncogenic stresses that lead to mammary transformation. This research project was initially a basic project with bench or in vitro data that would have been validated in animals models and with luck, finally, in human subject. However, as you will see in the next slide, very soon the bench results obtained could not anymore be interpreted or analyzed further with basic molecular or cellular biology tools. The goal was to understand if and how the EMT transcription factors were collaborating with oncogenic stresses to transform non-cancer mammary cells and compare these new oncogenic abilities or properties between these EMTTFs. The methodology chosen has been a screening where the 10 transcription factors will be compared in 4 oncogenic stresses conditions. The deletion of P10, of P53, the overexpression of EGFR, or the mutational activation of beta-catenin. That are all oncogenic stress found in breast cancer. So briefly, the virus contained the 10 transcription factor sequences were produced and infected in mammary cells that were already altered by an oncogenic stress. The original mammary cell lines did not possess oncogenic properties or transformant abilities, and adding this oncogenic stress was not enough to make them cancer cells. The infected mammary cells were grown in a soft agar plate that mimics the transformation process. And only cancer cells or transformed cells can grow in soft agar. After several days, clones were collected and amplified, RNA were extracted, and PCR were run in order to identify which EMTTFs have been overexpressed in the clones. In parallel, functional tests as soft agar, proliferation, and migration assay were performed to compare their oncogenic properties. Initially, we anticipated to analyze clones that have maybe 1 or 2 transcription factors overexpressed. However, because of a screening, we obtained clones that contain from 1 to 6 at 7 transcription factors per clone. And furthermore, the functional tests were showing that these clones presented a high diversity of transformant, proliferative, and migration properties. So a bigger picture of what was originally so was building up, and all this information have to be computed and analyzed. But how? And here is bioinformatics and biostatistics were the keys. And so we collaborate with a bioinformatician, and we gave her this simple Excel files that contain the combination of the EMTTFs in the column for each clone that are in the row and for the different condition, transformant and non-transformant, and for the specific oncogenic stresses. We ask a specific question, as we all the time have to do when we talk with bioinformaticians, one question at a time. Which EMT-TFs combination or signature were the most associated with a specific oncogenic stress? She thought that a tree-based model classification could help to answer our question. But at this time, so 10 years ago, more or less, the trees that we obtained didn't make sense for me, and I was quite confused on how to use them in order to answer this biological question. So I decided to go deeper in the tree classification process and understand the different steps of how they were built and how we had these complete trees. At this time, I only have Excel to help me, but I succeed to find a meaning, and also I succeed to create a new visualization that computes the number of the EMTTFs that we see on the circle, the colored circles, the EMTTF signature, and also that predicts the importance of the rank inside this signature. And so finally, also this visualization could predict the ability to drive mammary transformation represented by the width of the cycle. The triangle shows that their transformant abilities depending only on their rank if there were not other parameters altering these transformant abilities. And we could see that we can see on the visualization that the first EMTTF that have the strongest ability to predict the transformant properties of the cells will be on the top of the triangle with the first on the signature, and the one that has the least abilities will be on the bottom. However, when we put them together, we saw that we had a different signature according the transformant, according the oncogenic stress, and also inside the gene, this EMT signature, the transcription factors have an importance that were different. And so we could identify specific EMT signature according the oncogenic stress. This analysis was the beginning of understanding the bigger picture of the interaction and collaborations between EMTTFs in the context of a specific oncogenic stress during mammary transformation. In parallel to this project, the years of my PhD were also associated with the emergence of transcriptomic and genomic data online platforms. It was a decade now that the first human genome had been sequenced. Different institutes like the National Cancer Institute, the NCI, and the National Human Genome Research were joining forces to propose genomic, epigenomic, transcriptomic, protein, and also clinical data on cancer tissue with the TCGA, the Cancer Genome Atlas, also on cell line with the Cancer Cell Line Encyclopedia, and all that for the international research community with a free access. The possibilities to validate and understand the bigger picture of what we obtain from our bench results were now within everyone's reach, even the ones that didn't have any bioinformatics expertise. My bioinformatics curiosity, already initiated by my own project, were fed by my boss that wanted to use these new tools, and I took the challenge to learn how to use them. What I didn't know at this time is I will fall in love and be addicted to bioinformatics. The bench work, as maybe a lot of you know, where we can spend hundreds of hours trying to accumulate results and dispose them as a kind of puzzle pieces, were not anymore very exciting for me. And I largely prefer to spend the same amount of time resolving bioinformatic puzzles. So as I said previously, I was using Excel as the first tools, and Excel showed very quickly its limits, and I had to learn from scratch real bioinformatics and biostatistics software like R. So I didn't take a university classes, but already at this time and more today, there are excellent tutorials online that are free or not much expensive, and forums where I could and I continue to ask the dumbest question without any shame, and people are very patient to answer them. And so all this, the fruit of this effort led me to have different collaborations during my PhD, and one of them led to a publication in Nature Medicine. So I was quite encouraged by that. So after that, I left France and worked as a postdoc fellow in the University School of Medicine with Dr. Candil as a PI. Dr. Candil is an endocrine surgeon specialized in thyroid cancer. And so my research work was strongly driven by a clinical perspective. It was actually a very good training to never forget that my research project, even is driven by a strong curiosity, have all the time to have in mind, and that in the end, the research project have to help patients. And so today I would like to share with you two projects that show how much bioinformatics is a tremendous help to answer clinical and translational questions and problematics. So the first project is related to cancer and aging. So cancers are aging disease, and so more we get older and more the cancer incidence increases. However, the link between aging and aggressive cancer is not clearly defined, except in one type of cancer, thyroid cancer. Thyroid cancer is the only cancer where clinicians have established standardized prognosis marker using age as a marker component, because older patients, older than 55, have a higher risk of cancer-specific death. However, it is not the case for every older thyroid cancer patient, and it is crucial to identify which all patients will present a high risk of poor outcome and aggressive tumors. The new identified prognosis marker will assist clinicians to establish personalized and more accurate therapeutic strategies. First, for the high-risk patient with more aggressive therapies, and also for the low-risk patient with less aggressive therapies or active surveillance, where no surgery is proposed, but a regular controlled follow-up will check the evolution of these low-risk tumors. The active surveillance is the future of thyroid cancer, as a majority of these cancers are low risk, but consequences of surgery and treatment are high. So in order to discover this biomarker, we use the TCGA thyroid cancer and the GTEx thyroid normal tissue datasets. And different gene expression analysis were performed with a DEC2R package, so using the R software, in order to select which genes were the most significantly differentially expressed in this supposed aggressive aging cancer population. So we perform different comparison analysis between aging cancer with aging normal tissue, young cancer, young normal tissue, aging normal and young normal tissue, in order to select specifically the aging cancer gene. And we found 198. From this first selection, other DEG analyses were performed comparing the clinical and survival parameters, and we filter the 198 genes on 72 cancer aging specific, but also aggressive genes. As we focus on an aggressive phenotype associated with the aging process, the same analysis, but adjusting the parameters with age, were next performed, and the 72 were filtered as 48 cancer aging specific and aging dependent aggressive genes. Finally, as internal validation, the same analyses were performed only on the aging cohort to be sure that the genes selected continue to be associated with an aggressive phenotype in this aging cohort. And we finally define 23 genes that are cancer aging specific and aging dependent aggressive. A score computing the expression value and the beta coefficient of regression analysis of these 23 genes were calculated. This score validated the strong association of the 23 gene signature with aggressive phenotype and also with the poor outcome. The score powers to predict the aggressive phenotype. Of this poor outcome were estimated with the receiver operative curve analysis. The interpretation of the curve is the following. The area under curve AUC. The closest is to 1, and the strongest the predictive power to classify is. And so as you can see on this curve, the 23 DAG panel presented a very strong AUC, superior to 0.7 for most of the clinical parameters analyzed. Hence, this 23 DAG signature predicts aggressive tumor phenotype in the all thyroid cancer patient. We went further and we compare this 23 panel with abilities to identify the high-risk patient with the already standardized clinical markers that are used in clinic today. And so we observed that actually the 23 DAG panel presented a significant and strongest prediction that the AGE system, the AGCC system, when we were predicting the overall survival, but also, again, the ATA risk classification that predict for recurrence with a disease-free survival and progression-free interval. Hence, this 23 gene signature is a potential prognosis marker for aging thyroid carcinoma and also can be used complementary to the standardized prognosis markers. Finally, through a principal component analysis and hierarchical clustering, 2 subclusters of this aging cancer population could be identified: the less aggressive aging cluster and the more aggressive aging cluster. So we have discovered a 23 DAG aging panel that predict which all patients will present a poor outcome and aggressive phenotype. Validation in other databases and also in human subjects will be needed, of course. To conclude, this panel may lead to a more accurate personalized therapy and a better understanding of how aging aggravates cancer progression. So the second project that I want to share with you is related to the cancer process, the resistant process after BRAF inhibitor treatment in melanoma patients. So more than 60% of melanoma and also in thyroid cancer present a mutation of the kinase BRAF. The main mutation found is the V600E with the valine, the 600 amino acid of BRAF protein is replaced by glutamic acid. The mutation leads to a change in the protein conformation, making the kinase pocket being overactivated. The overactivated kinase leads to an increase of proliferation and survival of the mutated cells, and so create and drive tumorigenesis. The overactivated kinase, so drugs were designed to be competitive substrate, especially on this mutated kinase. And so under BRAF V160E inhibitor treatment, melanoma shrinks and patient will be in remission. However, or unfortunately, most of the patients will relapse in the next 18 months after the end of the treatment because cancer cells will have induced resistant mechanism against these specific drugs. So it is crucial to discover new biomarkers as gene panel in order to identify which patients have a high risk of recurrence. The new biomarker will help clinicians to adjust the therapeutic strategy by proposing a combination of treatment with the BRAF inhibitors and drugs preventing the resistant process, or by choosing another frontline therapy as a immunotherapy. So the project began at the bench where our team was looking to understand the resistant mechanism after a BRAF inhibitor treatment, because these mechanisms are quite still unclear. So we performed an in vitro melanoma model of resistance. We used, an in vitro melanoma model of resistance, and we performed a proteomic assay. Among more than the 80 different proteins, 18 were differentially expressed in the resistant samples compared to the sensitive one. Next, we were interested to see how these proteins were behaving in human melanoma-resistant models. In our knowledge, there were not protein data available online to answer our question, so we decided to look for transcriptomic data. In the GSC dataset repository, we found 5 datasets of human melanoma samples that were treated with BRAF inhibitors and present resistance. We focus on the first, the GSC 19898, has a discovery panel, and we validate our results in the other datasets. So first, we checked the differential expression of the 18 genes according to the resistance stage, and we saw that most were deregulated, but few presented a significant difference. However, when we run a principal component analysis in order to estimate the importance of the contribution of the 18 gene to explain the heterogeneity observed across the samples, we saw that the 18 gene could significantly differentiate the samples according to their resistance stage. With the pink samples represent the sensitive one and are in the left of the plot, whereas the blue samples that represent the resistance samples are the right of the plot. Furthermore, the genes presented a variety of correlation, as we see on the right plot, but also a variety of contribution to the PCR, with the red one having a high contribution, whereas the blue one having a lower contribution. And so, as we observed that all the 18 genes didn't have the same contribution to the resistance discrimination, we decided to filter them and select a signature that presents the strongest predictive power to stratify the sample, according to the resistance stage. To this aim, we use the supervised machine learning linear discriminant analysis methodology for every combination possible between the 18 genes. So 260,153 possibilities. All these combinations were tested, and the criteria of selection used was a ROC analysis with the estimation of the area under curve of the AUC. And so the combination presenting the highest AUC have been chosen. It was a combination of 18 genes, and this 18 gene signature presents a discriminatory strength of 0.98 as the AUC in the discovery dataset. Linear regressions and other statistical tests shown strong significant p-values, and other datasets used as validation tests presented similar results. Finally, a merge of the 5 datasets in order to obtain a dataset that have a high number of samples were built with the computation of the corresponding z-score for the individual previous datasets. And this merge shown also a very strong discriminative power of this 13 gene signature to predict resistance in human melanoma samples. The ROC curves illustrate the table, the previous table with the 6 analyses, the 5 dataset and the merge of the dataset. And we observe that all of them have an AUC superior to 0.7. The 18 gene score as previously for the 18th gene were also differentially expressed according to the resistance stage in all the datasets. And through a PCA, we could see that the 18 gene explains the difference observed across the samples because of the resistance process. So we have discovered a 13 GAG resistant panel that predict which melanoma patient will relapse after BRAF V600E inhibitor treatment. As previously said, validation in other datasets and human subjects are needed. To conclude, for this project, this potential therapeutic marker might help clinicians to accurately establish a personalized therapy. This panel analysis will improve the understanding of drug resistance mechanisms. And so today, I work full-time as a bioinformatician, finish bench work, and I work at the vet school of LSU at Baton Rouge with Dr. Kousoulas. Being part of the I am part of the LBRN as the Assistant Director of the Bioinformatics, Biostatistics, and Computational Biology Core. And I will be the liaison between the LBRN community and the Pan Biotech. It will be my pleasure to collaborate and help and mentor you for your bioinformatics or biostatistical needs. Thank you.
You're on mute there, Grant.
There we go. Thank you, Emmanuelle. We appreciate all of that information. We have learned so much from everybody today, and you're working in a changing world with changing data structures, and it must be a huge challenge. Thank you for delving deeper and giving us more understanding in what you're doing. I'm going to ask you a few questions that we've got in, and I'm going to get you a start here. What are the challenges in bioinformatics in relevance to personalized medicine?
On my experience, what I observed is there is the wish of clinician and researcher is to find the most optimized signature marker that can be used for the health problematic. However, bioinformatics have a diverse methodology to answer this question. And there is no one that is better than the other. So the challenge for me was how to choose the best methodology and how to justify the why I have chosen this methodology and not the other one. That was for
thank you.
A challenge that you haven't resolved.
What prepared you for this position, and what do you think other students might learn and master as they embark on their own bioinformatics analysis?
What was the first part of the question? Sorry.
What prepared you for the position?
What prepared me? During all the years, every time I have a new question, a new necessity on bioinformatics, I have to learn it. So it's a learning process. What prepared me for this position is during this my postdoc, particularly, I had the opportunity to collaborate with different persons that present me in the end, Dr. Kousoulas. And I think the fact that I have this background in molecular biology, in science research, where I understand the bench work, I understand the molecular process that we are trying to understand, it's an advantage to help by researcher or student for a project, because I have this balance between the molecular biology and biology context, but also know which kind of bioinformatic tools are available to try to answer the different questions. And furthermore, also, when you have this balance, it's maybe easier also to ask biological questions. I think sometimes what I heard when bioinformaticians were talking is it's difficult for them to have a clear question of what they need to answer with their tools. And so having this two expertise in a way help to have a clear question to be answered.
Very good, thank you. So what do you think, we have a lot of students on our calls, I know, what do you think students might want to learn and master as they embark on their bioinformatics analysis? What kind of advice do you have?
I think it would be interesting for them to at least maybe beginning to master some free software as maybe R, that is very easy, the ones that I use, on very basic statistic and bioinformatic tools has correlation analysis that can be used for different field on research, can be beginning to understand also statistic that can be used in very diverse field and are not very much specific for molecular biology or immunology or this or other things. So yeah, it can be helpful to have a kind of basic understanding of the bioinformatic tools that are available.
Very good, thank you.